Hi everyone, my name is Maria Pombo and I am a senior in the college majoring in psychology and minoring in cognitive science and computer science. Today I will be presenting my project on the perception of and appreciation for multiple vertical axes of symmetry, which I conducted as a research assistant at the GU Neuroaesthetics lab under the mentorship of Dr. Norbert Grievax. So a little bit of background here, we have Raphael's School of Athens and Van Gogh's Starry Night. The painting on the left, which is Raphael's School of Athens, is a great example of how vertical symmetry has been studied in the field of neuroaesthetics in the past. Symmetry is usually treated as a single feature, and a painting is either symmetrical or not. In this case, this painting would be considered symmetrical. However, in reality, symmetry exists in art differently, so Starry Night is an example of a painting that contains a lot of smaller symmetrical axes that are embedded into the painting itself. Previous neuroscience research has suggested that symmetry activates brain regions of higher order, such as the lateral occipital cortex. These brain regions tend to have larger receptive fields, which are represented by the circle on the left. This makes us wonder whether we can in fact detect multiple axes of symmetry at the same time. On the other hand, psychophysical research has suggested that we can detect symmetry within a small degree of the visual field, suggesting that we could detect multiple axes of symmetry. We therefore ask the question: can human subjects detect multiple vertical axes of symmetry? If so, how do these contribute to the aesthetic value of art? We conducted 3 psychophysical experiments and created 3 sets of stimuli. They're all dot patterns. So here the one on the left is completely random. The one in the middle contains 1 embedded axis of symmetry, which you can see over here, and the one on the right contains 2. So on the first experiment, we showed subjects an image with either 0 or 1 axis of symmetry and asked them if they saw 0 or 1. We manipulated exposure time and the distance of the axes from a central fixation point. The second experiment was the same as the first one, except we used stimuli with 1 or 2 axes, and we also manipulated the distance between the axes. In the third experiment, we showed subjects 2 images with varying numbers of symmetry axes and asked them which one they preferred. So here are the results of our first experiment, and the main result of our first experiment was that embedded symmetry can be detected very quickly in a small eccentricity, and these results replicate previous findings and served as a control of our own stimuli. And the graph here to the right shows that the greater the distance, the harder it is to detect symmetry, but even at 100 milliseconds and even 30 milliseconds, subjects could detect symmetrical axes at levels greater than 10 at short distances from the fixation point. Here are the results of our second experiment, and the main finding of this experiment was that in fact subjects could detect 2 axes of symmetry. There was a significant effect of time on detection of multiple axes of symmetry, so the greater the exposure time, the easier it was to detect a second axis. So a possible explanation for this finding is that symmetry is detected sequentially, so you first detect one axis and then detect the other one, explaining why you would need more time to detect 2. Another possible explanation is that receptive fields in areas like the lateral occipital cortex contain multiple units of symmetry detection. Lastly, here are the results of our third experiment, and the main finding here is that subjects have a clear preference for a larger number of axes of symmetry, and this can be seen very clearly in the graph to the right. And there could be a possible effect of time, and there were strong individual differences in our results, but it is evident that participants have a preference for more axes of symmetry. So based on the results of experiment 3, we got a lot of anecdotal evidence that suggests that maybe it was the shapes formed by the symmetrical patterns, like the one you can see here to the right, that made these patterns appealing. So we are in the process of running another experiment in which we embed these shapes in the random dot patterns. Some of these shapes are symmetric, some are asymmetric, and some patterns are completely random. Here are the sources I used for my presentation. Thank you for watching my presentation. Overall, I really enjoyed my experience as a research assistant for the GU Neuroaesthetics lab. I learned a lot about the field, a lot of technical skills, and I think the one thing that surprised me the most was that you could in fact use science to study art, do things that I thought were opposite beforehand.